this particular gene has this kind of an implication and then actually treat it without the drug. So that's the practical applications of the AI ML tools.
Good afternoon, everyone. Thank you for joining. And good morning if someone is joined from outside of India. And it's always pleasure having all of you for these sessions. To start with the introduction, myself Saritha, been in the industry for over 18 years now, primarily into the clinical programming and the biometric space. And I am your moderator for the day. And we have a very interesting topic today to understand the role of AI and ML in life sciences. And we have esteemed speaker with us, Raghavendra Gowd, who is the executive director with Pharmnxt Biotech. He's been in the industry for over 27 years of pioneering contributions to biotechnology, biologics, and molecular diagnostics across India and South Asia. A distinguished researcher from the Indian Institute of Science and an alumni of both IIM and ISB. He brings a rare convergence of scientific depth and strategic leadership. Thank you so much, Raghavendra, for joining us today and accepting our request to be our speaker. And I'm not yet done with introducing him. That's just a quick glimpse of, but there are lot more accomplishments of our speaker. So his career spans a transformative journey through the biotechnology landscape, leading impactful roles at GE Healthcare as a country head and later at Saritha as general manager for India and South Asia, where he drove growth across biologic manufacturing and healthcare infrastructure. As the global CEO of EPME Diagnostics, he led the development of next-gen precision tools such as EmfloDX, a rapid diagnostic kit endorsed by World Healthcare Organization for multi-drug resistant tuberculosis. Currently, as the executive director at Pharmnxt Biotech, he oversees global commercial operations, shaping the future of pharma manufacturing solutions. Beyond his executive leadership, Dr. Raghavendra is a dedicated ecosystem builder. He's an active investor, advisor, and a founding promoter of startups redefining biotech innovation, including Jenny Power RX, ATGC Biotech, XAT Diagnotech, Transcell Biologics, Platex Winters, EPME Diagnostics, and Jaitra Innovations. His portfolio reflects a deep commitment to next generation healthcare, genomics, diagnostics, and sustainability. His unique blend of scientific credibility, corporate excellence, and entrepreneurial mentorship positions him as a powerful voice in shaping the intersection of AI, education, and biotechnology. His story is one of innovation, impact, and relentless pursuit of excellence. He's also been recognized as one of the top 20 influencers in India's biopharma industry by Biospectrum, sharing the stage with industry icons like Dr. Kiran Majumdar Shah and Dr. Krishna LR. That's a great achievement, and congratulations for that, Raghavendra. His thought leadership has also been showcased in Economic Times, CXO Outlook, Global Edition, CNBC, ET Now, and other leading platforms. With that, I am very privileged to welcome Dr. Raghavendra for our session today to explore what is the role of AI and ML in the life sciences. With that, all over to you, Raghavendra. Thank you so much for joining us once again.
Yeah, Saritha, thank you so much for this opportunity to speak on this platform. Thank you for bringing that exhaustive introduction. So what I would do is AI in life sciences is very broad, very deep. It actually takes tons and tons of time. But what I've done is I've picked up few areas where I have been working and the portfolio companies that I'm working with are involved, and especially in the life sciences space. So that's what I'm going to present. And this is going to be more of a personalized stories and then how things can benefit not just the ecosystem, but also the life sciences is more related to the humans, the beneficiaries are the human beings across the globe. So that's how I'm going to bring in. So now moving on to the current topic today, as I've said, role of AI ML in the life sciences is a very broad topic, and it spans from drug discovery in the therapeutic space, in the diagnostic space, in the predictive medicine space. Apart from this, then you also see the AI ML being implemented in agri, veterinary, microbial, mammalian. So there is this large areas where AI ML has been its implications or its applications, and then this implications of AI ML in the lifespan. So I've just chose 3 or 4 topics where we are working and how we are able to use these AI ML tools. So the first one is, of course, on the core discovery space. We all know predictive models, light from your identification of the lead candidate, target identification, and then screening those candidates, trying to identify the right candidate for future. There are these lot of these predictive models. There are these, 2D, 3D models that are available, which actually helps you in the discovery space. And from there, clinical research, drug development, and manufacturing. And I'm going to present one slide on the manufacturing, how these AML tools are being utilized in the manufacturing space, and then the general areas of applications, especially in the patient side of the story. And we all know the advantages. The biggest advantage is it's fast. While if you want to just identify a candidate, it would take few months to years. That has been reduced to weeks to months, where you actually identify the lead candidate. And the scientific experiments that are supposed to be done, the vet experiments take immense time. And instead of getting into those vet experiments and doing a simulation model, also helps to reduce the time, effort, and money, and more importantly, the speed to market, what we call. That is one of the biggest advantages of implementing the AI tools in the discovery space, in the lead identification space. And then the other most important is, we all know in a discovery space or identifying a new therapeutic molecule, it takes minimum to millions to billions of dollars. And if you implement these tools, the observation is that it is actually going to reduce those investments that go into identification of the new candidate. Data points tells that it's about 55 to 60% of the reduction. And then the other most important is, this is again, from a psychology of the scientist. As you see the results quickly, then there is this huge motivation that, and you can work on multiple projects at the same time. And that's one of the biggest advantages of implementing the AI ML tools in the discovery and therapeutic areas. Now, moving on to the next slide, which actually talks about how AI ML modules have revolutionized the clinical space. As you can see on the first slide, the poor mouse says that only 92% of the drugs tested on me, 92% of the drugs tested on me, we fail. And apparently it's only 8% of the drugs that move into the next stage. Imagine when we say 92% of the drugs, imagine the kind of sacrifices that, we have to make, not just in terms of time, effort, money, but also the ethical issues around the animals and all those things. Now, imagine if you have a tool or a technology that gives you the precision for about 8% or 10% of the drugs, and then tell, out of these 100 candidates that you're testing, only 8 would probably move into the next stage of your discovery process, and you don't need to sacrifice the animals. That's what FDA is driving these days, what is called as a modernization act, where even the FDA says that you don't need to sacrifice the animals. Try to find out alternative models that can help you to screen the candidates, move to the next phase. And that's where, one of the companies in Hyderabad is pioneering this work, where instead of the mice, instead of the animal models, this company is trying to use the stem cell models. And this is across what we see, the using the stem cells models, you actually get a humanized data. While it is a mouse is a mouse is a mouse, I mean, obviously it is not mimicking the humans, but here when you use the stem cells, these are, human derived stem cells. So obviously what we call as a microphysiological models, human microphysiological models give you the data points which are almost similar to the humans. So that's the biggest advantage. And we all know the importance of the stem cells. So imagine if stem cells are employed or deployed instead of sacrificing the animals. The 92% of the poor mouse is talking. Imagine all that screening is done on the stem cells. It's a huge savings, right? And that's what this is, how this technology is being used. So what, and most of us know stem cells have got this capacity to get differentiated into various cells. And then there are these technologies or, the patented tools or the processes that helps to develop to or differentiate these stem cells into various organs of interest. So you can actually differentiate these stem cells into cardiomyocytes, neuromyocytes, or skin cells, or even neurons. So depending on the drug candidate, for example, if you're trying to develop a candidate for, cardiac, then you don't have to sacrifice an animal, but develop develop the stem cell model, which is differentiated into cardiomyocytes, and incubate your drug candidate with these cardiomyocytes, and you would get almost the same parameters what you would have got in a mouse model. So now, similarly, drugs that you want to develop for neurological disorders or something like skins, or there is this exhaustive list of things that can be done using these stem cells. And actually, as you can see here, these are the differentiated models that have been developed in the lab. So these are, if you can see, full-blown nerve here, or a full-blown skin test, skin grafts, or even the cardiomyocytes here, cardiac cells. So this is the importance of using the stem cells instead of using the animal models. What we do here, or what's been done, is these molecules of various origins. It could be biomolecule, it could be small molecule, it could be biologics, it could be proteins, or it could be monoclonal antibodies, vaccine candidates, which are supposed to be therapeutics or the preventive kind of vaccines. These are subjected to these differentiated stem cells, and you do various kind of studies on the stem cells. You have those all kind of markers that you're looking for. They are available on these surfaces, and then you expose your drug to these stem cells, you incubate them, and you start working on the parameters that would give you the data point. So for example, the phenotype, you would see if this drug candidate is actually killing the cells. If it is killing the cells, this means that this drug will not be moving forward. This is toxic, right? And then there are these models, so differentiated models that can tell you how this drug is not going to move forward. And similarly, the genetic expression, the outcome of the incubation is subjected to various, to identify the genetic signatures, transcription factors, the receptors, and then you put them into the AI models. Already there are these models that have been trained. There is this huge amount of database that is created, and these data points that comes out of these experiments is subjected to that. And then you get a scoring that will tell you that if this drug candidate can move forward or not. If it is not moving forward, what is the next thing that we need to do? Or if it is moving forward, what is the outcome? For example, if you want to study the neurotoxicity, if you want to study the immunogenicity, or some of the safety concerns, neurovirulence, and in fact, this can also tell you the potency of the drug that would be effective on a candidate. So this is the advantages of employing or deploying the AI ML tools. So obviously there are these mathematical or the machine learning tools that have been developed, and then the data that comes out of these wet experiments is subjected to these models, and then you get the outcomes of this. So now here, the outcome of the whole thing is that you're reducing the cost, your time to market is going, and then more importantly, the ethical issues around the animal sacrifices is also reduced. And I'm sure some of you who've seen how expensive it is to get the animals, rear the animals, and then experiment on them, and then there's also a cost that is involved. You're actually reducing the cost and ensuring that your candidates are more or less working on humanized models. That's the biggest advantage that we see when we employ AI ML tools in the clinical trials. Now moving on to the second one, this is more in the manufacturing where we have developed this model. If you see the biomanufacturing space, whether it is a vaccine manufacturing, or if it is the monoclonal antibodies, cancer drugs, or the biologics, or even the insulins for that matter, when we actually manufacture, so there is a biology that is involved. So unlike chemical synthesis, where it's more of reactions in a reactor, but in a biopharma manufacturing, it is a fermentation process. So you have a clone that has a gene of your interest, and that gene of interest is propagated. So from a small bioreactor, you move into the large scale bioreactors, which varies from 1,000 liter to 2,000 liter, and it takes about 1 to 14 days. So what it means is you inoculate your bioreactor with the clone. It would take about 14 days to grow and give you the yield that you are looking at. And in that 14 days, you need to provide the media, which has all the nutrients, your growth factors, your glycogen, sugar, aeration, oxygen, and everything. Now, how your AI and ML tool is helping the industry is, so what you do, this is a process, and there is this data that gets generated. For example, you're doing 1,000 batches of this production, and you have the data. And it is not that every batch that you inoculate or every bioreactor that you inoculate with your culture is going to stay for 14 days and give you. The success rate is less than 50%. So this means either the cells die on the third day, but you will not even know that the cells have died till the 14th day, till you actually look out, but you have not got the yield that you're looking at. So what we call as a sweet spot, which means that you get the maximum yield on a particular day of your fermentation process. Now, when you actually use your database where you have seen that one bioreactor has given you the best harvest on the 13th day, while there is another parameter while there is another batch that has given you the best outcome on the 10th day. But you don't know that it is on the 10th day you've got, but you still run the bioreactor for another 4 days, expecting you'll get it. But biology is biology. After a point, the cells wouldn't grow. On the 10th day, if you've got the maximum yield, it is better to harvest the batch on the 10th day and start working. So you're saving the time, effort, money for the 4 days. So that's an advantage. So the sweet spot, what we call in the biopharma industry, especially in the biomanufacturing, that is what gets identified by the database that has been generated, which tells you there is a particular pattern that gives you, when the harvest is at the full of its maximum yield. So when you overlay the data, you will be in a position to tell that, this batch has a contamination and, should be aborted maybe fifth day. So this is how you save the manufacturing cost. So the biggest advantage is you will be in a position to identify the sweet spot at whatever intervals it is coming out. So that is the biggest advantage. So you save a lot of nutrients that goes in, a lot of factory time, and overall this is going to be bringing efficiency and then the cost effectiveness, and every day cost a lot to the company. So if you're able to save those 4 days and get your maximum, this means the company is benefiting on the costs that are going to get incurred on those 4 days. So this is one of the advantages, and this model has been developed at GE. We have a lab in JF Center in Bangalore, and this is what we have developed, where this is being deployed in the biopharma industry, where we're trying to use AI ML models, what we call as a digital twin. And many biomanufacturing industries today are being benefited out of this digital twin. Now, moving on to the third most important applications, which is my favorite. So if you see, very simple, there is this client, you collect your blood from the client, and then subject, you extract your DNA and then subject the DNA for genetic sequencing. And then that sequence looked upon the markers. So on the right side, you see there are these a lot of markers that you see here. And these markers have been extensively published. So you can see there are markers for high blood pressure, there are markers for migraines, respiratory allergies, gastritis, diabetes, renal, degenerative diseases, mood swings, anxiety, mental disorders, cholesterol disorders, CADs. So these are set of genes that have already been identified, and this is available in the public domain. And this is what being used as predictive models as well as the precision diagnosis. Now, how this is going to help, and I'm going to give you practically how people are benefiting out of this. For example, on the left side, you see how the diabetic patients are benefiting out of this, the model that has been created. So there are these genes, HNF4A, GCK, HNF1A, and IPF1, HNF1B. These are these, and some of the genes that have been picked up. And on the right side, you see the effect of the mutation. And I'm going to give you how the patients have benefited out of this. So generally, what happens is, when someone walks into a clinic with a high blood sugar and the HbAc, HB1AC levels. So the tendency of the doctors is that they would put people on metformins or the sulfonylurea drugs. But when you do a underlying one more analysis of genetic analysis, what it tells you is, we had a patient where this gentleman was put on the metformin for more than 7 years, but is not in control. What we have identified is that he had a mutation of GCK. This means that it is just the diet that needs to be controlled, and this gentleman wouldn't need any medicines at all. So, once this gentleman was identified and, the genetic profiling was done, when this was identified that he had a mutation of this particular gene, and then when he was put on the diet control and, other lifestyle management, then, we were lucky enough to identify that this particular gene has this kind of an implication, and then he was actually treated without the drug. So that's the practical applications of the AI ML tool. So you have the markers database, and then there is this genetic sequencing that's being done, and this genetic sequence is, put on these AI ML models, and then this is an outcome, how a patient is benefiting out of this. And similarly, someone might not need an insulin, but then he is put on to the various drugs, and someone who is on a metformin can actually respond better. So what we call as a pharmacogenomics, so whole genome sequencing, pharmacogenomics, and then the nutrigenomics. So some of the advantages, while I'm not an expert on these things, but I would give you my perspective and how our organizations are benefiting using the AI ML models. And similar is the case with the cardiac mutations as well. So you see here, there are these particular markers and then the effect of these mutations, and it actually helps the doctors to predict better, understand the underlying root cause, and then helps them to narrow down on the medication. And that's what we have seen, and both the founders being the physicians themselves, they have been extensively using this. And today, this company has pioneered this, and then they are the backend for many of the leading hospitals in India. Not just in India, but globally, they have their presence, and then they have used the AI ML models as one of those precision diagnostics as well as personalized treatment. And here is an interesting story. So you see here, so I have been a very chronic migraine patient right from 10 plus 2. I've been a chronic patient of migraine. It's one of those severe kind of situations. And I've been taking all the drugs that are related to the migraine from maybe 10 plus 2 or my graduation. So when I've got my DNA genetic profiling done, and then what we have identified, and this is a very interesting pattern that we have seen, when my genomic sequence was subjected to the migraine markers, which are 220, you can see the score, right? You can see. So this means, the score tells me that I'm not supposed to have migraine at all. But then what's the causing factor, right? And I've been not knowing the root cause. I have been on the migraine medicine for more than 20 years. Now the doctor, the physician, identifies that the migraine is not because of the migraine, but then they looked at my respiratory profile. Here is an outcome. So there are these 3 genes what they have analyzed, and then look at my scoring. These are, you have the highest impact on the respiratory gene. So now the correlation is that since my respiratory profile has an issue, when I'm subjected to any of these allergens, that triggers the sinus, and sinus triggers the migraine. That's the connection between these two. And once this has been identified, that migraine is not because of the migraine genes, but it is because of the respiratory problem, or some of the genes that are allergens that are getting impacted by this. The treatment regime is very simple. What has been suggested is, whenever I feel allergic or whenever I feel get into these kind of situations, either a steam inhalation or a simple antihistamine. And you'll not believe, this is almost like 2 years I've not taken a migraine tablet till date. And that's what I say, how I have been a beneficiary of the new age technologies. And there are a lot of people, especially the breast cancer patients. We've screened a lot of these breast cancers, and both in terms of the pharmacogenomics, what drugs suit a particular mutation, and also the dosage pattern. That's been one of the biggest advantages of this. So this is where I say how AI ML has been very personalized applications. You see the applications of AI ML in your day-to-day life, and that's what I wanted to bring in. Like I've said, I'm not an expert on this, but wanted to bring in those applications where you personally feel, yes, this is going to be a game changer. This is going to bring in a lot of advantages, not just in the therapeutic space, diagnostic space, but across the patient population. This is something that I wanted to highlight. And then also the other one, what we do is a risk prediction. The same company also does the risk prediction, and it's the same, no-brainer here. So you take your blood, there is this genetic pool that is available. There are these genetic conditions that are associated with. Then there is this profiling of the medical conditions is done. They correlate the data, identify which marker is impacted, and then obviously we have tie-ups with American, some of the leading colleges and some of the leading databases across the globe, especially Mayo Clinic, American College of Molecular Diagnostics, and many of those we have associations. And what this predicts is, can actually predict the scoring on the diabetes, cardiovascular diseases, your cancers, some of these nutrigenomics profiling, and everything can be done. So that's one of the advantages what we have identified. And if you see, like I've said, 3 major advantages of AI in this model is, one is improve the accuracy and efficiency or efficacy. The other one is, early disease detection. Risk assessment using the genetic profiling is something that we have done for many of the patients, and then we have seen the impact of genetic profiling, and then how that can be used as a base for further treatments, getting into the right treatment, getting into the right dosage, getting into the right pattern is what we have observed. The other biggest advantage is decision making for the medical professionals is something that we are enabling. So while we know a particular set of drugs would work, but within that particular set, what has the maximum impact is what we have identified, and then that's what we do. So this is something that I wanted to catch up. While I know it's very exhaustive a topic, Any questions, will be more than happy to answer, Saritha and the audience.
Sure. Question from Rajnikant is, you want to ask it by yourself, Rajnikant?
Thank you, Raghavendra, sir, and Saritha facilitating this one. But however, right, I have experience in, right, processing the data from the most of the global pharmaceutical industry, where I see that, sir, I see the continuous manufacturing, the facilities were very less across the globe. I see that Sativa is the one, right, maybe where the G have the much of its facilities is a continuous manufacturing facilities, but it is very less, right, the spread across the globe. So that how can, right, IT companies can reach out the technology lack that, industries, and then benefit them. So how can we reach out to them, sir?
I mean, if you want to reach out to Sativa, I can connect you because I was the
No,
managing
no, no,
director.
sir. And Sativa is well ahead.
I
So
was,
I'm not
I
saying.
was the,
I'm just giving a reference that like Sativa, there are a lot of biomanufacturing companies, small molecule companies, are yet to implement this, the advanced the technologies within their manufacturing process. Be it, I would say that in India we have close to 2,500 manufacturing API facilities, whether it's a dose API packaging manufacturing facilities,
yeah,
are like running with a very old traditional. You see the Sigachi case, right? So it could have been identified.
yes.
So by implementing our historical production process, right, prevented, and then somehow this is not the social benefit is not happening. I don't know what is actually blocking these industries to embrace these new technologies. So maybe, right, we are working, IT companies, and we're working on a day-to-day job, and we are not getting an opportunity, right, to think in that extent. To reach to that level, definitely we require ranks in my hierarchies, right? We may not do that. So this is an opportunity that I'm asking. I don't need to follow any hierarchy. I'm just putting my question.
One of the industries which is slow in adapting the AI ML tools is the pharma and biopharma. While I was running GE, so I was a part of the the design teams in Bangalore. I was a cross-functional team, so I was closely working with locomotives. I was working with aviation. I was working with couple of those things because there are some of the digital twin model that you have seen is actually that we have picked up from an aviation industry, and
Right.
similarly there is what we call as a bio insight which we have picked up from a healthcare industry and we have tried to implement in the biomanufacturing. So what does bio insight mean is, for example, there is this cath lab going on and then there is this catheter. I mean, especially when you do an angiogram, what happens is there is this catheter which is injected into the body, and then you try to scan the blood vessels for those the blocks, right? Now imagine if the catheter is in the body and machine conks off. It's
Yeah.
possible to take it out, right? For example, if the engineer has to come, it takes a lot of time. Now that's where concept called as an insight, which is a tool that has been developed where remotely an engineer can control the machine and then he can actually take out the catheter so that the patient is safe. So obviously patient population is very important. But that's something that the biopharma industry has picked up. And then what we try to do is, for example, all the biomanufacturing industries are not in the cities. They are like 30 kilometers away or 40 kilometers away. So, for example, while we sit in the city, the manufacturing centers are in Genome Valley, which is like almost like 40 kilometers from here, right? Imagine an engineer has to go. It's very different. Now, what we have done is we have developed this a little more, and we call it as a bio insight, where we actually take the machines into control. Sitting here, I can actually take the control of the machines. Look at what's happening in the manufacturing process, identify the issues. If it is solvable, solve it. Or what we call is, divert broth, because when you want to manufacture, you're actually putting almost a crore worth of raw material into it. If you're not identified, it just goes as a waste. So that is where we've been strongly advocating that you need to implement these tools. So our observation is both pharma and biopharma are the late adapters of these AI, ML, or even the new age technologies, because there are these markers, there are these FDAs, EMEAs, and then there are these regulatory approvals that are needed. Any change that you make in a manufacturing is a big process. You have to report it till your FDA levels, and Saritha would add because she also comes from the similar background and she understands why it takes a lot of cost, time, and effort for anything you want to bring in a change in any processes, because all it has to go all the way till to an FDA approval. So you need to apply for a change control protocol, and then it's a big process. So that's where the lag is. But keeping in view the advantages what the AI, ML, or even the new age tools are bringing in, there are these new setups that are coming up, are implementing these.
Fantastic. Thank you very much for the elaborate response, Raghavendra, sir.
To add on to it, yes, there was a resistance. I don't say there is currently, because I think even in the industry, everyone understands that AI and ML is in place, and we cannot say because of the regulatory we cannot implement it, right? Because we see the advantages, we see the efficiencies what the technology brings in, and there are a lot of organizations who are heavily investing on how we can implement these technologies in expediting the overall drug development process so that the medicines can be brought much earlier to the people. With that, I also see there is another question. The question is, are there any use cases or startups working on the clinical trial space, like the recruitment or the site management, etc., with the AI and ML usage?
Yeah. There are a lot of companies, and if you see, the startup space is gone very high. In fact, so what we've observed is the number of startups have gone very high. So from a 2,000 to it's today, I think 5,500, and we're expecting that to reach to 10,000 very soon, because that's another area that government is funding heavily to bring in the innovation. Where Prime Minister Modi ji is telling that we're known as a pharmacy of the world, but can we be known as an innovation of the world? We can we bring in these innovative tools in the life sciences? Having said that, there is this huge population that is actually getting into the startup space, and then there are many startups which are in this space as well. In Hyderabad itself, there are about 14 incubation centers. Similarly, Bangalore has a large population of incubation centers, and then Pune is another space where you have these incubation centers, and then of course Gujarat is picking up. So I think there are these startups which are working on these areas, so you might reach out to them and see how that can be done.
Yeah, to add on your point, and you're absolutely right. Definitely, everyone are adapting to the technology.
Yeah.
And then, Kumar, to answer your question, I'm not suggesting or recommending only one region, but then my recent experience in one of the external conference, we've got an opportunity to visit T-Hub. Because the conference was being held in T-Hub, and then I must say I was very amazed with the kind of ecosystem what has been built. And then, I'm not saying again Hyderabad is the only place, but then I was amazed because that was my first experience seeing the kind of ecosystem and the kind of support what government has put together in order to support the startups, and then seeing the different ventures, the different startup organizations, and then it's amazing. I can't, it's, I go out of words while I have to express that, but then it's very amazing. And then there are a lot of young entrepreneurs who are coming up with lot of problem statements and they're trying to solve. Because me being in the industry for somewhere close to 2 decades now, I have seen lot of my colleagues who were working with me have, because we have seen what are our real challenges, and then they are working on some of the solutions as entrepreneurs now with their startup companies. And then there is lot of high possibility and potential even from the corporates to adapt to these technologies. Everyone understands that if we are not adapting to the technology, if we are not bringing these technologies into the in-house, we are not going to be sustaining in the industry. That's where people are ready to implement and adapt to the technologies. That's where you see the data management, or even if you call the site management and all, now people call it as data scientist. That does not mean the technology is going to take away the jobs, no, because that's another point which always comes in. But then it's a mixture of how can we adapt to the technology having the human in loop. Morning I was watching one of the talks, and it was very beautifully said that technology cannot take away the jobs, but then people who do not use the technology would get impacted. Human has to be in the loop, and then the kind of thought process, the creativity and different outside aspects of it what human can only think can only done by the human, and technology cannot do everything for you. It only enhances our abilities to do things more efficiently, more faster than what we are doing it in the current time period. I'm not sure if that adds up, but then there are a lot of pieces. I mean, if you see the overall clinical trial drug development, every aspect of it, the technology is getting enabled to make it much faster and improve the overall life cycle of getting the medicines to the humans. Okay, any other questions from the team? There is another question. While we see this gene therapy, Raghavendra, but then we do see some of therapeutics like rare diseases where we really do not get even the participants to do that, and that is something which gets a little, and then we don't get the medicines for rare diseases. I would like to understand from your experience if this gene therapy kind of thing has been explored for the rare diseases where you really do not get the participants, but then you can develop it and do the experimenting of it in order to identify the molecules, Raghavendra.
So stem cells definitely has this potential, differentiated what we call as a humanized microphysiological models did show a promising results. Various therapeutic candidates, especially some of these rare diseases, something that people have picked it up, and then you see, you do see, but obviously, you can only narrow down to a certain extent, but you cannot actually be obviously it's a treatment, right? I mean, you need to have the volunteers for the final phases. So
Yeah.
but at least till that stage you can get it, and then there are these, some of the people who have been impacted are becoming the volunteers, while for others you get a lot of these volunteers, and then there is this, guidelines around it. But for rare
Yeah.
diseases it's a little bit difficult. But
Okay.
stem cells, your CAR-T, chimeric antigen receptor therapies, and your gene therapies, personalized treatments are showing some positive results, Saritha, and we have very good examples. In fact, in one of the associations I was presenting on the impact of the new age, especially the cell and gene therapies, and
Mhm.
how it is going to be very, very effective instead of those regular therapies. Yeah.
Okay, that's great to hear. And I totally agree because the precision medicine is taking a very
Correct,
promising thing.
correct,
And
correct.
then, that's very much where people are looking, and even looking at the real world data, the variables and all, that becomes very promising in order to expedite the overall process. Looks like we are good, and thank you. I really appreciate all your time and sharing your insights, Raghavendra. And thank you to all our participants. Thank you for joining and being very active participants. We really appreciate your time. And thank you so much, Raghavendra. Thank you. Thank you, everyone. You have a great day until we see you next week with another interesting topic.